Good to see that so many of you made it this morning after the conference event yesterday. This is the second session that is co-organized by the Sonderforschungsbereich 1066 in Mainz. It is on polymeric materials for cancer immunotherapy. And in the session today, we'll be focusing really on the immuno aspects of nanotherapies. This afternoon, we will have another session in which we look at different types of diagnostic and theranostic approaches to personalize cancer immunotherapy. So please also come to that session. It will be at 2 o'clock in the main hall. It is my great pleasure to introduce the first speaker, who is Dr. Volker Mailänder from the University Clinic in Mainz, who has done very instrumental work in understanding how the protein corona contributes to cell uptake by different types of immune cells. And maybe you can also say a couple of words about the SFB to introduce it to a broad topic.
Okay, thank you very much, Twan. So the idea that came up in Mainz was that nanoparticles should be especially interacting with immune cells because they are about in the same range as viruses or cell debris and are taken up by, for example, by dendritic cells, which represent the most important checkpoint in order to determine what is an antigen and what is not an antigen and how can you vaccinate against a tumor. So I'm working at the university clinic. That's where I come from, basically, from transfusion medicine, selecting stem cells, and the nanoparticles you will see, they come from the Max Planck Institute for Polymer Research. So why should we care for proteins and nanocarriers? Well, first we want to use proteins in order to target these nanocarriers to specific cells, like dendritic cells. And what we've learned over the years is that there is a lot of that these nanoparticles, as we make them, as the chemists make the surface and make them a wonderful, nicely designed surface, once they come in contact with blood, they are covered with a bunch or with a whole new coat with a protein corona. That's something Dawson has mostly pointed us towards and has given us a hint why sometimes targeting does not work. And even some of these proteins may do a mistargeting, so they will still be functional in terms of that they bind to cellular receptors and that we get then cellular uptake, unspecific uptake we don't want in the first place, and also we get a different biodistribution on a microscopic level. So maybe there's another solution. Maybe we should, the first thing that we were looking at, because all these macrophages, all these dendritic cells took up the particles in an unspecific way, but we wanted to target specific subtypes of the dendritic cells. So the question is, how can we get rid of all these proteins and the nanoparticles? How can we do something like a stealth effect to them? And the most applied polymer for that is polyethylene glycol. And I will show you in the second part that we can also use absorption of proteins and use them in order to target cells specifically. But let me start with the polyethylene glycol, the stealth effect for this part. So the idea was that instead of having all these proteins on the surface, that we try to avoid them, and that we decorate our nanocarriers with stealth polymers, which is in this case polyethylene glycol. This is the most known polymer for using this. And the idea and the paradigm, the dogma, so to speak, was that once you put that on your particles, you will find no or nearly no proteins on the surface, so they all stay away from your nicely designed nanoparticle, and they can no longer do this mistargeting and change the behavior. And then you would, for example, get no cell uptake if you have no other targeting moiety on the surface. In this study, we used two different types. We used a very nasty, so to speak, nanoparticle, which is polystyrene. There are a lot of things stick to it. So once you coat that with polyethylene glycol, and we use two different chain lengths, or we used here another polymer which was designed in the Max Planck Institute for Polymer Research, which is polyethyl ethylene phosphate. It resembles a little PEG, but the difference is that PEG is biodegradable, and so much should have less concern than PEG in the end. So this was the first experiments we did. The question was, how much of the proteins do we find on this? And when you look into other publications, then you see, well, you still find a little bit of these proteins on that. So you could say it reduces protein absorption substantially, or you could say it does not completely hinder protein absorption. Sorry. And we looked at it, the second part. So the question was, can we then see a difference in cellular uptake? And there is really, if you use nanoparticles and incubate them in plasma, you really get no uptake. But if you do that, and you can do that for a few hours with the cells, if you just take the naked nanoparticles, if you don't put plasma into your cell culture, then you see there is still a lot of uptake here. And it looks like more like really that the protein is the one who is doing the job in terms of non-uptake. So what's that protein? For the P particles, we can clearly see that the most abundant protein with about 80% is clusterine, apolipoprotein J. For the PEC, there are two of them. There is clusterine and there is apolipoprotein A1, and both together make this stealth effect. So basically what we did, we changed the paradigm from no absorption to specific absorption of some proteins, mostly of the lipoprotein complex series, and this then gives this no uptake effect in the end. The benefit of that is that once we have different materials here, we can just check for the protein corona, which can be done in a short time, something like a day or two. You can know what the proteins that stick to your surface are and which are not sticking there. So people have been trying to make particles where nothing sticks, and I would say there's a good test for that. So take 3 different cell lines, take also raw cells, are monocyte, monocytic cell line, macrophage cell line, and maybe also primary cells like leukocytes, and then look if they have, if you can change uptake by protein absorption or not. And if this is not a case, then I would say then we have really particles which do not have a protein corona, which would be very, very much interesting to do further targeting with them in a, so to speak, in a pristine form. Do that with and without the proteins and test uptake. So the second thing is, how can we use absorption? Maybe we can use the targeting moieties we put on the surface deliberately, so to speak. And that's the standard approach. You use usually EDC NHS chemistry on a carboxylated nanocarrier. This then binds the antibody, and this is usually done on a pH 6.1 and monotonic gold. And his chemical colleague, they decided that I will do a, so to speak, a negative test for that. They said, well, we will not do the chemical couple, we'll just absorb it, but we use the same buffer, also pH 6.1. And then nothing should stick to it. And once you have your lysed dendritic cells, then the particle which has nothing on it should not stick to the band where the protein is, but the particle with it should be then sticking there. We call this a Western blot. And then you have a secondary antibody detected, and you can really see the antibody itself is detecting the CD63, which is a marker for, which is a antigen, which is on the surface of dendritic cells. And then you have the covalently and the absorbed antibodies, and they both bind an IgG, which is not directed against, does not bind all the particles themselves. They don't just stick to the membrane. So, but this was all done without proteins, and so of course the question was now, can we then also, when we do the chemical coupling, how much then is human serum affecting the uptake? And what you can see is that you get a good targeting if you have no serum, if you have no proteins. If you have 10% serum, there's a reduction of that, and if you use 100% human serum, which is rarely done in the literature, but which can be done if you do pre-incubation, then you see there it's really very much reduced. So the covalent bond antibodies, they are covered efficiently in serum, they are shielded. So what happens if you just absorb them? Does this still work, or does it just go off? Well, that was the interesting point, that it even works better, and that even with 100% serum, you see a reduction. There is an influence, definitely. There's a little bit of shielding, but it's very much better than anything else, and gives you still a very good targeting in the presence of serum. And is this something we could have expected? Well, when we do ELISAs in sim-linked immunosorbent assays, a lot of time we also use whole serum or whole plasma in order to do the analysis. So in this case, it works with a flat surface. So there is also a good chance that it could work with a curved surface. Why we were a little, or Manuel was definitely lucky, because when you look here, here's pH 6.1, and you see there's the highest absorption, the highest detection for the antibody in this case. And you can do this, of course, with the anti-CD63 antibody, so with a monoclonal antibody. Here we used mouse IgG, which is a combination of many subtypes. But it also holds true for this, that we have less uptake, that we have less absorption of the protein in the basic regime, but in the acidic regime we have more, and there's an ideal for this carboxylated one. And if you look then for an aminated surface, amine functionalized surface, then you see you have to go then you have to go very, very low with your pH, even to pH 2.7, in order to see some uptake. This here means that we have also other IgG instead of IgG2A absorbing at pH 11. So why is there better targeting? And this is just a short reminder that you have the stem part of the antibody, so to speak, which is also some functions. Some functions we may not want, but you have definitely what you definitely want. Why you put it on it is the FAB region, the antigen binding region. That's why you want to have that's where your targeting comes from. So we were looking if we have similar amounts of the protein of the antibody itself on the surface, and we did this with secondary antibodies against the Fc part. And what you can see is that you have, for the covalent and the absorbed ones, you have about the same amount. No matter if you have the monoclonal or the polyclonal antibody, then you have the same amount of Fc sticking out or detecting. And and what you then, if you detect the FAB part, then you see there is a big difference between the covalent and the absorbed ones, that the covalent obviously cannot be detected so much, that the FAB part cannot be detected as well as before when you have it covalently bound and compared to the absorbed ones. And does this also hold true for a longer time? Yes, it does. Here for this aminated surface, you can see that it holds at least true for 24 hours. It reduces about 30 to 40% the amount, but there's still something there for the carboxylated one, which is even with which we started. That's even worse, but still you have something like 40% of your antibodies which you can find after 24 hours, which is probably long enough to do a targeting in an organism. So why should absorption be better? Why do we think that? When we look at our data, then we think that in some cases, even due to chemical coupling, even maybe coupled like that you have your FAB parts directed both to your carrier surface, and you have nothing left in order to bind to your target, the CD63 antigen in our case. But of course, in most times, you will have at least one of them sticking out and being able to interact with the antigen. And the best thing, of course, is when you have both sticking out. And why should that be? Well, there are other papers that have determined that at low pH, the Fc part denatures, and then probably this looks for a hydrophobic surface or for a surface where it can attach to, and polystyrene is definitely a good marker for that. We have also done that with others, with hydroxyethyl starch or with protein capsules, and you can also use this in order to do this absorption and to have a good targeting afterwards. So the take-home message for today is antibodies can be absorbed to nanocarriers in such a way that they withstand the protein corona formation, that they can still do the targeting. If you want to do that as well, you need to look for different pH and look where the most and the best targeting occurs. Can we do that on all nanoparticles? Well, certainly not. But we have done it on some of polystyrene, hydroxyethyl starch, and also on albumin particles. So that works at least for these. If it does work for liposomes, for example, is not yet clear. With this, I would like to thank all the people on the list, especially, of course, the first three. Johannes, Manu, and Toni Gold, I did the work with the targeting. Susanne Schöttler did the polyethylene glycol part. And then, of course, the SFB and all the other people who supported that and will take that further on. Thank you very much.
Thank you very much, Volker. Professor Henrik.
Puzzling me a little bit. So if you are doing targeting, we also think you need the covalent bond between the targeting ligand, the antibody, whatever,
Yes.
and now you claim now you should not do that, it should be simply physically adsorbed. But the risk is, if you then do that and you inject it, and before you reach the target tissue of the target cells, you get exchange of proteins, etc. So then the targeting ligand will be gone before you reach the target site. Can you comment on that?
Yeah, but that's exactly what we've shown, that you can do even for 24 hours, and you can still find antibody in it, and you can still do the targeting. So we also looked if they are still targeting to the cells in this case. We have not done that till now in animals. So we have to look for organisms, definitely.
Yeah.
And do I say we have to do that absorption? Well, it's a fast and easy way to do it. But definitely it would be much more elegant to use the Fc part to more specifically in order to do a chemical coupling. So that would be much more preferred also in terms of pharmaceutical, that you have a pharmaceutical product in the end and not something a little bit undefined too, or you even get rid of the Fc part, because some cells will just recognize the Fc part and you probably don't want that if you talk about macrophages or things like that. So that would be, there's definitely room to improvement, and I'm sure that the products will not, in the end, will not have that absorption of the antibodies. It's a good way to test if your targeting works on a lab scale, but if you have something that you want to sell in the end, I would go with a chemical coupling. Yeah.
Thank you. Volker, if you adsorb your antibodies to the surface of the nanoparticle, could it be that you sort of adsorb several layers of these antibodies, and that they will then stick out of the protein corona to make them more accessible for the target?
What we have always seen is that, and what we have calculated is most of the time that when we wash the particles like we do, and we wash them hard, then we have a single protein coat on the surface. And also if we have the immunoglobulins on them, we probably don't have more than one coat. If you have proteins which interact with other proteins, like antithrombin-free together with other partners, then you may have several layers of it. But IgG, because we only pre-absorb IgG, so I don't think that there is a good interaction between two IgG molecules. So I would think that this is really a one layer.
Question in the back.
You can also use the microphones on the sides if you want.
Yes.
This reduces at least a little bit the jogging here.
Yeah. Thanks. So I was just trying to figure out the binding mechanism of the proteins on the nanoparticle surface.
Yeah.
And you said that the binding is more efficient when you bring the pH to a very low values.
Okay. Yeah.
But for me, it's I couldn't understand this, because when you bring down the pH, especially if you bring down the pH below the PI values of the proteins, and the proteins are mostly positively charged, then I would say they look for negatively charged surfaces, but not hydrophobic surfaces. The proteins, they are most hydrophobic. Sorry. They are most hydrophobic when the pH is the same as the PI.
I think you have two partners here. You have the surface of the particle, and you have your protein or peptide. We also did that with interleukin-2, for example. And there you can also do that, this absorption, and that I still stick to it. Besides that charge difference or charge, that you were looking for, most of the proteins that absorb, or let me put it that way. If your theory of ionic interaction would be correct, then you would find of a positively charged particle, you would find negatively charged, so to speak, biomolecules. And there are a lot of papers which show that the PI of the protein is not a decisive factor for absorption. So what we think, and what I think a lot of other people think, is what is the other interactions, van der Waals or hydrophobic interactions. And as there's a good hint that the proteins, or especially the immunoglobulins, they unfold a little bit, and they unfold hydrophobic patches, especially in the FC part, so in the stem part. So then this will look for a surface where it can attach to, and get, so to speak, can get rid of the water to interact with, but it tries to interact with another surface.
And in this regard, I wonder if you do the adsorption at when you adjust the pH to the PI value, what does it happen? I would suppose the proteins they're more hydrophobic, and then at this point the adsorption would be more efficient.
You're right, but you still have also to have to nanocarrier surface in order to be also then really hydrophobic. And if you have a carboxy functionalized or otherwise, or an amino functionalized surface, then you will also have charge there. So there will be always an interplay, or you will have two determinants: the surface, the nanocarrier surface, and the protein itself. And we just did that we just did the experiment, so to speak.
Okay, I have a more general question. So, do you know if viruses have a protein corona in vivo? And if yes, would it make sense to pre-coat particles with something like a protein corona before you inject them, either locally or intravenously, to really mimic the mechanism by which the immune system recognizes viruses?
People have been trying to infect hepatocyte cell lines in vitro with adenoviruses. And they were not successful. And why were they not successful? Because we always use serum, which is a lot of times even heat inactivated. And the adenovirus uses complement factor 10 in order. So it basically, you have the virus, it absorbs a complement factor 10, and then it uses this as the entry point into the hepatocyte. So if you do that, if you really work then with non-heat activated, or if you add again complement factor 10, then you can get into the cell.
Is that something people are pursuing for immunostimulation or immunotherapy, to pre-coat with factors that are known to promote uptake by immune cells?
That I would know of.
Might be an interesting strategy for the SAP, or at least consideration. Yep. Good. Last chance for a question? If not, let's thank Professor Mailänder again.